Have you ever wondered if we could modify the blueprint of life itself? Welcome to the cutting-edge world of gene editing, a technology that equips scientists with the power to tweak an organism's DNA. This could mean adding, removing, or altering genetic material at specific points within the genome, effectively letting us manipulate life's fundamental building blocks. Just pause and ponder for a moment. Imagine the possibilities if we could rewrite the genetic code. That's the power of gene editing. So how does gene editing work? It's like using a molecular scissors. Picture this. Your DNA, the blueprint of life, is a long, winding strand. Along this strand, scientists pinpoint a specific location they'd like to alter. They then employ enzymes, nature's own tiny machines, to cut the DNA at that exact point. Here's where it gets really interesting. Once the DNA is cut, scientists can add or remove genetic material. Want to insert a beneficial gene? You've got it. Need to take out a harmful one? Consider it done. This isn't a rough cut and paste job, though. It's a precise operation performed with meticulous care. Because of this precision, gene editing allows for highly specific modifications. We're not just reshuffling the deck, we're customizing the cards. The potential for medical breakthroughs, new treatments, even cures, is immense. It's a process as intricate as it is revolutionary. Now, meet the game changer in gene editing, CRISPR. Short for clustered, regularly interspaced, short palindromic repeats, this mouthful of a term has revolutionized the field of gene editing. But how does it work? Think of it as a molecular GPS, guiding tiny scissors to the exact point in the DNA that needs editing. CRISPR utilizes RNA molecules, known as guide RNAs, to navigate the complex map of our genetic material. These guide RNAs are like the voice in your GPS, dictating the turn-by-turn directions to your desired destination. Once the guide RNA has led the scissors to the correct location, it's time for the cut. The enzyme Cas9, acting as our molecular scissors, makes the precise cut in the DNA strand. This cut allows scientists to either remove unwanted genes or insert new ones, effectively rewriting the genetic code. CRISPR is like the GPS system for our molecular scissors. It's a revolutionary tool, driving us into a new era of genetic exploration. But with great power comes great responsibility. This sentiment rings particularly true when we consider the vast potential of gene editing. This revolutionary technology opens up a new world of possibilities. Imagine a world where genetic diseases, from cystic fibrosis to muscular dystrophy, are a thing of the past. A world where our food crops are more resilient, more nutritious, more bountiful. However, as we contemplate the potential of gene editing, we must also grapple with the ethical implications. The power to rewrite the code of life is not something to be taken lightly. Unintended consequences could arise from even the most well-intentioned edits. For instance, a tweak meant to increase crop yield might inadvertently harm local ecosystems, or an alteration intended to cure disease could have unforeseen side effects. Furthermore, there's the unsettling possibility of misuse. In a world where genetic perfection is attainable, who gets to decide what constitutes perfection? Could we inadvertently create a new form of inequality, where those who can afford genetic enhancements enjoy superior health and abilities? Gene editing holds immense promise, but it also poses profound questions. As we stand on the brink of this new frontier, the decisions we make today will shape the future of life as we know it. As we delve deeper into the realm of gene editing, we must tread carefully, ensuring that our actions today do not compromise our future. So, what does the future hold for gene editing and CRISPR? As we've delved into the intricacies of gene editing, we've seen how CRISPR, a revolutionary tool, has the potential to rewrite the very fabric of life. This powerful technology has already made waves in the world of science, and ongoing research points to even more promising developments on the horizon. Imagine a world where we could eradicate genetic diseases or engineer crops to withstand harsh climates. That's the power of gene editing and CRISPR. But as with any powerful tool, it comes with its own set of ethical implications. It's a delicate balance between harnessing the potential of this technology for good, while ensuring we respect the natural order of life. In the hands of responsible scientists, gene editing and CRISPR could herald a new era in medicine and biology. It's a future that's exciting and a little bit daunting, but most of all, full of possibilities.